Welcome back, guys. We are covering more nervous system content. Today, we are primarily discussing action potential and what happens on a cell in a cell-to-cell level, especially these neurons, as far as how they send these stimuluses and these impulses through the nerves to eventually reach the cells that they need to activate. So, let's talk about that. Let's go ahead and get into that discussion. So first, before we get into that, we do need to discuss some things: irritability versus conductivity. This is stuff that we actually saw similar in muscular system. In muscular system, we saw irritability versus contractility. Contractility was how well the muscle contracts. Conductivity is more how conductive is the nerve or the neuron in order to transmit the impulse to other neurons, muscles, or glands. So just think of a conductor. A conductor this is stuff you should have learned in other science classes, right? But it's a device or a something that is going to be attractive to heat or electricity. It's the opposite of an inductor, right? Inducting and conducting, all that fun stuff. Okay, now we got to get into the meat and potatoes of this. Let's go ahead and talk about how a neuron is excited. And when we mention exciting a neuron, we're basically saying that the neuron is able to achieve action potential so that it can send the stimulus to the next cell. So at first, the cell is just at rest. That's what we expect, right? The cell at rest, it's relaxed. When the cell is at rest, we actually call it polarized. Now, a couple things that make the cell polarized. Number one, what ions are found. If you've never taken chemistry, you need to know this. Na is sodium. K is potassium. So we need to know those two. Sodium is on the outside of the cell. Potassium is on the inside of the cell. How do we summarize that? Well, there's an easy mnemonic for us. The mnemonic is salty banana. Why salty banana? Well, salt is made up of sodium. It's also chlorine, but Na. And bananas are rich in potassium. So salty banana. Salt on the outside, banana on the inside. You'll never forget it. So because of the way that these ions are interacting with each other, the inside of the cell actually has an innate negative charge. So it's a net negative. We don't need to dive too deep into the chemistry as to why is it negative. Don't even worry about that, but just know it's a negative charge. Inevitably, a stimulus is going to travel through the cell. As it travels through the cell, it's going to excite the neuron, causing sodium to rush in. That rushing in of sodium is called depolarization. Depolarization activates the neuron to transmit an action potential. That's what we're expecting to see, right? Which is the nerve impulse to the next cell. It is an all or nothing response, similar to the muscles, right? Remember in muscular system, we mentioned how when a muscle contracts, it's all or nothing. Similar process here. The impulse will conduct itself down the entire axon. Now, after that's done, you need to return to normal. So to return to normal, you utilize repolarization by potassium diffusing out. So you do need to repolarize inevitably. Now, in order to move sodium and potassium, we actually have a pump that helps this whole process out. The good news is the name of the pump is simply sodium potassium pump. Now, in most cases, diffusion is all you need, but sometimes you need to move these ions against the concentration gradient. What do we mean by against the concentration gradient? You're moving it against the normal flow. So diffusion is typically high to low concentration. Using these pumps, you're moving things from low to high concentration. But in order to do that, you need ATP. So this sodium potassium pump is actually an example of active transport. This is all stuff that hopefully you've seen in biology as far as passive versus active transport. Okay, now moving on from here. The resting potential. So we talked about at rest how the cell is negatively charged. What we do need to recognize is that at that negative charge, it's negative 70 millivolts. So we do need to know the number for this. So at rest, it's considered negative 70. Here we see those gated channels. Pretty straightforward, right? We're able to see what that they open and close to allow ions to flow through. This is important. So when depolarization is happening, what we need to pay attention to is, as you see here, a small patch of the membrane, which is on the neuron, will become depolarized. If you slow depolarization down enough, and this is happening at a very, very fast speed, but if you slowed it down enough, you would see that the vast majority of the neuron is at rest, and then when the stimulus is hitting it, that's the only part that flips. I like to think of it like a copy machine. When you're copying a piece of paper, the laser in the machine is scanning the paper bit by bit by bit. And as it's scanning, that's what's being copied over, right? Similar situation here, except instead of a laser, it would be obviously the stimulus transitioning through the neuron. Here we see pretty good rendition of what's actually happening. We have our resting state, sodium on the outside, potassium on the inside. You have your depolarization, which is going to be the sodium rushing in. That's also going to cause action potential. Here you can see how it causes a spike in voltage. Then you're going to have repolarization, which is when potassium pumps out, right? And then finally, hyperpolarization. This is a step we haven't learned yet. Hyperpolarization is just pumping even more potassium out in order to try to return the charge back to a resting state. Don't have to worry too much about this, but this is simply saying how fast this process is, right? You see that at rest, everything's obviously starting off, but then the stimulus moves through. And by the time the stimulus goes through and the cell returns to rest, that's typically 4 to 5 milliseconds. Very, very, very quick. Finally, saltatory conduction. We talked about this in the previous video a little bit, but the idea here is that when the signal enters the neuron and it enters through the dendrites, when it finally reaches the axon, the stimulus is going to jump from myelinated region to myelinated region. That jumping process drastically increases speeds. Here's what you can see. Electrical signal jumps from node to node along the myelinated axon to get up to 30 times faster. Now, that's everything I wanted to talk about in the notes itself. Let me go ahead and pull up a graph to help us out here for a second. We can just go ahead and use this one. Perfect. So in this context, what we see is what we were discussing, but this is a much more clearer graph. On the x-axis is time, on the y-axis is voltage. It's always in millivolts because this is a very small amount of charge, right? Step one, you're at rest. While you're at rest, you're at negative 70 volts, right? Millivolts. You'll receive a stimulus, which is that dotted line. Sometimes the stimulus is not powerful enough, so you actually get what's called failed initiations. Once it crosses the threshold, which is negative 55 millivolts, depolarization is going to kick off. It's going to cause a big spike in voltage all the way up to positive 40 volts, millivolts. After action potential occurs, you need to return to normal. So repolarization is going to occur, but it actually dips down to roughly negative 90 or so millivolts. Then to turn back, it's also called the refractory period, but you can call this hyperpolarization. You're going to dip back up in order to reach negative 70 again. This process we need to be comfortable with. So one last thing for practice while we're doing some practice today, right? Let's go ahead and just label this action potential. I'm going to actually turn this computer flat so I can draw a little bit better. So regarding action potential, step one was always resting. / polarization. Knowing this, what does the cell actually look like? So the cell, if this is the axon of the cell, right? The cell is going to have a ton of sodium on the outside and potassium. It wants to switch over. There you go. All right, the blue pen might not be working well for us. Let's see. There we are. Potassium on the inside. Because of how this works, that ends up generating a negative 70 millivoltage charge. Next, we can move on to our next step, number 2. And that's going to be depolarization. What's happening in depolarization? Well, that sodium that's on the outside is starting to flood its way into the cell. The depolarization also only occurs because of, we'll use this teal color for now, or we'll just use green. This depolarization also occurs because you have a stimulus traveling through the neuron. So I'll just color code for us in this green color. That is the stimulus coming from the previous cell, right? And the charge is now positive 40 millivolts. Okay, so now we flipped it to positive. Next, let's go ahead and create a new whiteboard so we have some space here. Step 3. Step 3 is going to be after action potential. So that's actually going to be repolarization. Repolarization. What's happening in repolarization? Here. The cell has now a ton of sodium inside and potassium. You got to get rid of this sodium and potassium. But first, we're going to try to pump out potassium. And we're going to try to pump out potassium with the help of a particular pump. Just use a light blue for now. So we're going to use this particular pump. I'm going to write inside of this ATP, indicating that you need energy for this. And we're going to use that pump to pump potassium out of the cell. So just for color coding purposes, in that light blue, this is the sodium potassium pump. The charge has now successfully gone down to negative again, but we're at negative roughly 90 millivolts. That's too negative, so we need to go one more step. Hyperpolarization. So what's happening in the cell? Oops, it's a little too close. What's happening in the cell? Let's take a look. Basically, you still have some potassium left inside. You're going to collect all that potassium and pump it out. By pumping out even more potassium, you're going to regulate the charge back to resting charge, negative 70 millivolts. Now, this seems, if you've been following along, this shouldn't be too terribly difficult as far as like, oh, okay, I think I understand this. But there is something to address here. Notice how at start, right? I'm going to go back to this real quick. At start, we had negative 70 millivolts and we had sodium on the outside, potassium on the inside. But when we concluded, we're back to negative 70 millivolts, but we have potassium on the outside and sodium still on the inside. Technically, there is one last step. We don't have to worry about writing it down or anything, but it's basically diffusion, where the sodium is going to move back on the outside and the potassium is going to move back on the inside. But the end goal of this is that we return to rest, which is negative 70 millivolts. Okay, so I appreciate you guys checking in. This can get a little bit dense, so do your best to try to practice this and review this. Obviously, if you need any help, let me know and I'll assist you accordingly. But I appreciate you guys checking in. We'll see you on the next one where we talk a little bit more about some other neurotransmitters and some diseases that affect the system. Have a great one.